In 1990, researchers in northern Italy began tracking 829 people. They followed them for nearly 2 decades. Every 5 years, they measured what these people ate. 146 different nutrients, verified every death with medical certificates and autopsies. At the end of the study, one finding stood out above everything else. One single compound, found in foods almost nobody eats anymore, showed the strongest inverse relationship with dying from any cause. Stronger than vitamin D, stronger than omega-3, stronger than magnesium, and here is what made this finding particularly significant for anyone over 50. The levels of this compound fall dramatically inside the human body after age 60, right when it is needed most. Most physicians never test for it. Most people have never heard of it, and yet the evidence connecting it to cellular repair, energy production, inflammation control, and muscle health after 50 is now published in some of the most respected journals in biomedical science. Welcome to MindBody Files. Today we are decoding a compound called spermidine. What the research says about its role in cellular aging. Spermidine is a naturally occurring compound called a polyamine. Your body produces it internally. You also obtain it through food, and every cell in your body depends on it, for growth, for repair, and for survival. In your younger decades, your body maintains adequate spermidine levels naturally. Cellular repair runs efficiently. Your internal cleanup systems operate at full capacity. Recovery is faster. Energy is more stable. Inflammation is lower, but after 50, and accelerating further after 60. The body's internal production of spermidine begins to decline. Research from the Brunick study, published in the American Journal of Clinical Nutrition, tracked 829 adults aged 45 to 84 over nearly 2 decades. Adults, followed for over a decade, found that those with the highest dietary spermidine intake had approximately 30% lower risk of all-cause mortality and 32% lower risk of cardiovascular death. Even after adjusting for lifestyle factors, this is not preliminary research. This is replicated, large-scale epidemiological evidence. So what is spermidine actually doing inside your cells? And why does its decline after 60 matter so profoundly? The answer comes down to 4 biological mechanisms. Signal 1, autophagy, the cellular cleanup system. Autophagy is your cell's internal cleaning crew. It identifies damaged cellular debris, dysfunctional mitochondria, misfolded proteins, accumulated waste products, and breaks them down for recycling. As you age, autophagic efficiency naturally declines. The cleanup crew becomes slower, less thorough, less effective. Cellular debris begins to accumulate. Spermidine activates autophagy by working through 2 specific pathways, inhibiting histone acetyltransferase and activating AMPK, the cellular energy sensor we decoded in our insulin episode, a landmark 2024 study published in Nature Cell Biology, revealed something particularly significant. Fasting triggers an immediate increase in the body's own spermidine biosynthesis, and spermidine acts as a necessary key to initiate the autophagy cascade. Without it, the process cannot fully proceed. This means that if your spermidine levels are declining after 60, you may be getting significantly less autophagic benefit from intermittent fasting than you would with adequate spermidine present. The cleanup crew is waiting, but the key that unlocks the door is missing. Signal 2, mitochondrial health, your cellular energy factories after age 50. Research shows humans lose 30 to 50% of functional mitochondria. Mitochondria are the energy factories inside your cells. When they decline, cellular energy production declines, recovery slows. Cognitive clarity fades, physical stamina decreases. Damaged mitochondria also actively generate excess free radicals and inflammatory signals, adding to the chronic low-grade inflammation that accelerates aging throughout the body. A 2024 study from the University of Basel on human neurons found that spermidine increases ATP production, cellular energy currency, improves mitochondrial membrane potential, and reduces free radical levels. It achieves this by stimulating mitophagy, the targeted removal of damaged mitochondria. In other words, spermidine does not just support energy production. It removes the dysfunctional factories and creates space for healthier ones. Signal 3, inflammaging, the silent inflammatory cascade. We decoded inflammaging in a previous MindBody file, chronic, low-grade systemic inflammation that builds quietly over decades, driving cardiovascular disease, metabolic dysfunction, and neurodegeneration. Spermidine directly addresses this cascade through 3 specific mechanisms. It inhibits EP300, an inflammatory gene program. It modulates NF-kappa signaling, a key inflammatory pathway, and it clears the cellular debris that continuously triggers immune activation. Each of these actions reduces the background inflammatory signal that accumulates when cells are not being adequately cleaned and repaired. When spermidine levels are adequate, the inflammatory hum quiets. When they decline, it grows louder. Signal 4, muscle stem cells, sarcopenia and recovery. After 50, muscle loss accelerates, not simply because of inactivity, but because of changes at the cellular level in the stem cells responsible for muscle repair and adaptation. These are called satellite cells, muscle-specific stem cells that activate after exercise to repair microdamage and build new muscle tissue. A 2024 study published in Cell Discovery found that older subjects had significantly lower spermidine levels in these satellite cells. Restoring spermidine levels improved satellite cell activation and provided a plausible cellular mechanism for combating age-related muscle loss. It is important to note that these findings are from animal studies. Human trials are still needed to confirm the specific effect, but the direction of the evidence is consistent. Adequate spermidine appears to support the cellular infrastructure that keeps muscle repairing. 4 mechanisms. 1 compound, autophagy activation, mitochondrial renewal, inflammatory quieting, muscle stem cell support. The question is not whether spermidine matters after 60. The question is how to restore it. And the research points clearly toward food, not supplements. A 2023 study found that taking purified spermidine supplements at 40 milligrams daily for 28 days did not significantly change serum spermidine concentrations, raising serious questions about supplement bioavailability. Until large clinical trials confirm otherwise, the evidence favors dietary spermidine as the primary intervention. This is the MindBody Files Spermidine Restoration Protocol. Step 1. Add wheat germ daily. Wheat germ is the highest concentrated food source of spermidine currently identified in the research. 1 to 2 tablespoons added to yogurt, oatmeal, or a smoothie each morning provides a meaningful and consistent dietary input without requiring any significant lifestyle change. Other high spermidine foods to incorporate across the week: nat2, fermented soybeans, one of the richest sources available, shiitake mushrooms, mature aged cheeses, broccoli, cauliflower, and green peas, spinach and other dark leafy greens. Important note, anyone with celiac disease or a wheat allergy must avoid wheat germ entirely. The other food sources on this list remain fully accessible. Consistency matters more than quantity. Daily exposure over months and years, not 2 weeks, is where the benefit compounds. Foundational foods of the Mediterranean diet. This is not a coincidence. It is a biological alignment. Legumes, whole grains, fish, vegetables, olive oil, and fermented foods collectively provide not just spermidine, but the full nutritional environment that supports cellular health, cardiovascular function, and metabolic resilience after 50. You do not need to adopt a specific diet overnight. You need to shift the pattern gradually. 1 Mediterranean meal per day is a meaningful start. 2 is a significant intervention. 3 is the biological environment the research points toward. Step 3. Combine fasting and movement. The 2024 Nature Cell biology study established something clinically significant. Fasting triggers the body's own internal spermidine biosynthesis. Movement activates the overlapping cellular pathways spermidine supports, particularly AMPK activation and mitophagy. Combining dietary spermidine with a 12 to 16-hour overnight fasting window and 2 to 3 weekly sessions of resistance training creates what the research describes as a complementary anti-aging architecture. Each element amplifies the others. Dietary spermidine provides the substrate. Fasting triggers the body's own production. Movement activates the cellular machinery that uses it. This is not a complex protocol. It is 3 signals working together, restoring a system that has been quietly declining. Before implementing any of the dietary or fasting protocols discussed in this file, please consult your doctor if you take anticoagulant medications such as warfarin. Wheat germ is high in vitamin K, which can alter the efficacy of blood thinners, manage diabetes with insulin or sulfonylureas, have active cardiovascular disease, or are under oncological treatment. The epidemiological evidence shows no link between dietary, spermidine, and increased cancer risk. It actually shows protective associations, but those with active cancer should consult your oncologist before any dietary changes. This file is educational. It is not medical advice. Your doctor knows your specific situation. We do not. After 50, your cells are not failing. They are operating with less of something they depend on. And biology, as we have said before, is forgiving. The protective benefits of spermidine are long-term phenomena. You will not feel different in 2 weeks. What you are doing when you add wheat germ to your morning meal, when you build toward a Mediterranean pattern, when you combine fasting with consistent movement, is not a short-term intervention. It is a decade-long conversation with your cellular biology, and your cells already know how to respond. They have simply been waiting for the right inputs. Spermidine connects 4 of the most critical biological systems we have decoded on this channel: autophagy, which we covered in our zombie cells episode, AMPK, which we decoded in the insulin episode, inflammaging, which has its own dedicated file, and mitochondrial health, the energy system at the root of fatigue after 50. Every file we publish connects to the others, because your body is not a collection of separate problems. It is one system, sending coordinated signals, and every signal has a response. If this file gave you clarity, subscribe and turn on notifications. Every week, we decode one more signal your body is sending, calmly, precisely, and without alarm. Comment below. Had you heard of Spermidine before this file? Let us know. I will see you in the next file.